Hi, everyone. For those of you that don't know me, I'm Thomas. Those that do know me, I'm sorry. I am a technician here within mass spectrometry, and I'm in charge of structural proteomics here at the LMB. For those of you that didn't see Kat's talk, this is what the mass spectrometry facility at the LMB looks like. So Kat Franco is our commander-in-chief on the end there. Sue is the lab manager, and then Frida. And I do technician work within the facility, and Tom Smith is the newest to the facility, and he's in charge of all of the bioinformatics that we do. And he's a really good guy, and just drop him an email if you've got any questions about any sort of bioinformatics. And he's super helpful, really knows his stuff. So that's me. And I do the structural proteomics. I also do the protein intact mass analysis. And I've also aged terribly in the last 3 years since that photo was taken. So essentially, the thing that keeps me up at night when it comes to mass spec and proteomics in general is this idea of it's all a bit of a numbers game, really. So I'm not a biologist, so it's a bit weird for me to be talking about this to biologists. I'm hardly a scientist, quite frankly. But essentially, we're interested in what's in the cell. That's what we do. That's what we're looking at. And roughly 25,000 genes code for roughly 20,000 proteins. So there's 20,000 potential proteins that can be expressed at any one time. But what we're actually interested in is knowing the genome and knowing the transcriptome is one thing, and what proteins are present, but we actually want to know what's interacting with what. And I'm sure that's what you actually want to know too when you do your experiments. And what we want to know is how many interactions and what those interactions are. The fact is that there's lots of interactions, but nowhere near as many as there could potentially be, right? So this is a really good paper. I recommend everyone have a look at it at some point soon. And essentially a guy called Matthias Mann, who's like an absolute proteomics genius, kind of sat down, did thousands and thousands of protein tags, thousands and thousands of pull downs on those. And in 3,927 proteins they detected within yeast, they got 31,004 interactions. So sort of a nearest neighbor of about 8 proteins per protein. So every protein was on average interacting with about 8 other things. Well, that's kind of interesting. But the number could have been that. If everything was interacting with absolutely everything else, that's how many zeros would have been on the end of it, which is a lot of zeros. And that shows you quite how specific proteins are, right? We don't just do one pull down and get like a Pangeic mass of every single protein interacting with every single other thing. So how do we narrow that number down, and how do we actually work out what is interacting with what? And another note here that we're not even talking about proteoforms, which is what actually keeps me up at night, which is the idea that post-translational modifications completely change how a protein interacts with another protein. And there could be millions of those. So we're just building more and more complexity on top of complexity. So this becomes something that's very challenging. The number for a human cell, not a yeast cell, that's the maximum number of interactions that we might have, which I've decided is probably slightly difficult. We need to narrow those down, right? So what we can do is we know that not everything is acting with everything else in every possible permutation, but how do we go about solving that? And that's where structural proteomics comes in. So we're sort of this thing right at the bottom. And the reason I sort of open with this introduction is because I find it pretty interesting, but also because we talk about proteomics a lot, but structural proteomics is a bit of a different beast. We're not just trying to look at 20,000 different proteins and try and get an idea of what's present. We're trying to look at the different structural interactions. So instead of the number of searching, our search space being 20,000, our search space is that number underneath it. And it completely changes how we've got to approach these problems. Now, within the facility here, we do quite a few different experiments when it comes to structural proteomics. The two main ones are hydrogen exchange or hydrogen-deuterium exchange. and cross-linking. I'm going to spend essentially the whole talk today focused on cross-linking, how to do cross-linking better, and hopefully trying to persuade you guys to run some cross-linking experiments at some point in the near future. I'm sure, again, you're all biologists. This is pretty obvious to you. This is a pretty simplistic protein complex within a membrane. Normally, how you guys work out what's interacting with what is you do some sort of tagging experiment. You add a tag to one of the proteins in the complex. This is how Matthias did his stuff. You have some sort of tag, and then you do some sort of pull down an IP. And what Kat was talking about yesterday is that you can start with this tag protein complex. You can pull it down. You then just put it in a little Eppendorf, do the digestion, trypsin digestion. That gives you a series of peptides. Now, these peptides we can run on liquid chromatography into a mass spectrometer, and that can tell you exactly what proteins are present. That's super useful, right? We get protein complex interactions through positive identification of the proteins that are present. We don't tag one protein, do a pull down, and end up with 20,000 of those proteins, right? We only end up with 5, 6, 7, 8, maybe 20, maybe 60 if it's a really big complex, but we always end up with a much, much, much lower number of proteins than could be potentially expressed in the entire cell. So that's super useful. But what's annoying is the moment we do this step here, right? We take your lovely intact protein complex that you spent years, optimizing and doing all of that stuff, so you know it's active, so you know it does everything. You digest it and you lose all of that useful information, right? So all of a sudden, we've removed information that we had, and what we're trying to get around is how we can change that. So is cross-linking a potential answer there? The answer is yes, otherwise this would be a really short talk. So what we do is we take a small covalent cross-linker and we add it into that protein complex. And what this does is hopefully add a small linker into the one protein to another protein within the regions that they're interacting. We then take this, do exactly the same process again, not quite, we do some changes, but essentially for what you guys need to know is exactly the same protein process again, but now we've got a complex that's cross-linked together. We then digest and we end up with peptides, still trypsin digested peptides, but now that they've got a covalent cross-linker between two different peptides, hopefully from two different proteins. So now we can work out what's interacting with what and where they're interacting. So we've retained structural information. It is a slightly more complicated experiment. The reason it's slightly more complicated is now instead of just searching for one peptide within a single mass spectrum, we're looking for two peptides, and that not only has, normally a peptide has two fragmentation ladders, we're now looking at two peptides, which then has 4 different fragmentation ladders, which makes the processing a lot more complicated. But also we can get fragmentation ladders that contains the other peptide, so it becomes 8 fragmentation ladders, which is a bit of a nightmare, but we can get around that. And we're also, our search space is now, it becomes that second number. We're looking at what could potentially be interacting with everything else. What I'm going to talk about as a lot is how we can reduce that number as much as we can, and that's where you guys come in to hopefully get the best data possible. I'm going to skip right to the end now. What does this data actually look like? It can be kind of hard to picture in your head. But essentially, this is just some experiments that we've run. I've had to sort of clean up this data a bit, so can't tell you what it is. But essentially, that's what a dimer interaction might look like. We've got the smaller protein on top interacting with a larger protein on the bottom. This is what a tetramer might look like. So actually, let's do it. Any guesses to why we've got no cross-linking in this first region here? Go on. So what that bit is the bit that's sat in the membrane. So that's sat in a membrane, and the other bit is sticking out of the membrane, and those are all systolic proteins there that it's interacting with. So we're looking at cross-links of a tetramer there. So you can see that we get dense linking between different areas, and we can see what's interacting with what. And then that's an allotomer. I didn't bother counting the proteins, but there's a lot of proteins in there that are interacting with a lot of different things. Now that's super useful when you get to this size of complex, because obviously there are some proteins there that might only be interacting with 1 or 2 other things, and other things that are interacting with lots of other things, and that can really help narrow down what proteins are interacting with what regions are interacting with what, how we can narrow down our search space. And when it comes to you guys running, say, point mutations, things like that, you can actually work out what proteins are interesting. So that's super useful. But what are we doing? What is cross-linking? And it's pretty simple in principle. So when you want to run a cross-linking experiment, essentially what you do is you add this thing here at the top, some sort of activated species, and it's got two active sites on it, so it can react on one side and then react on another side. And we normally have something, this is an NHS ester, and that can react with any amine, and obviously on proteins, the most active primary amine is one on lysine. So it reacts with one side, and then you end up with this active species. We still have an NHS ester present there. So we've got protein 1 with the lysine reacted to a crosslinker. And then if protein 2 is in close enough proximity, another lysine on protein 2 comes in, and then we've got a covalent link between the two proteins. So we can then take that, digest it in a normal mass spectrometry workflow, and then analyze what we've got interacting with one another. It's pretty simple, but we do have some things that we need to consider. So firstly, reactive amino acid availability. This is obviously for a di-NHS ester like this, lysine is the most common thing that we're going to be using. What is important to remember there is if you're doing some things like intermembrane complexes, you likely won't have lysines within the hydrophobic regions, or much less lysines in the hydrophobic regions than other regions. So that's something that we need to bear in mind. Potential hydrolysis of the crosslinker, so the speed of the reaction of the crosslinker that you've used. So it reacts on one side, so you've got an activated NHS ester, that can be hydrolyzed. So you've got a crosslinker that's actually, this one here is now useless. So water has hydrolyzed off the other end. If the reaction is too slow, we end up with just a load of dead end crosslinks, and we don't get the cross-linking information that we want. The other big one is a lot of the times we're interested in inter-protein interactions. We're interested in what protein is interacting with what other protein. But obviously, you know, it's a covalent crosslinker. It doesn't know what is a different protein or not. So if you've got two lysines on the same protein in close proximity, they will form an intralink. We normally get about 10 times the number of intralinks than we do interlinks, because it's more favorable for a protein to react with itself, because normally itself is in closer proximity than something that isn't itself, which kind of makes sense when you think about it. So we get a lot of, oh, I've just dropped my mic. We get a lot of intraprotein interactions and less interprotein interactions. So what else are we interested in? We often need to look at the crosslinker concentration. So just generally what the density of the crosslinker will be on those lysines as we add it in. We also need to bear that in mind, because if you add in an awful lot of crosslinker, you can get dissociation of the complex, because obviously you're reacting lysines up, and a lot of the time they have something to do with the interactions between two proteins. I've got distance and dynamics here. I have lots of arguments with reviewers about this, and there's lots of papers that go back a really long way. We can use what's called zero-length crosslinkers, things like EDC, which you often use for peptide coupling. These work great, and you can use crosslinkers with larger arm spaces in, and people have made crosslinkers of really massive lengths. What's really interesting about that is it does make some difference, but it really doesn't make as much difference as people tend to think it does. So if you use a zero-length crosslinker, that doesn't necessarily mean that that lysine, in the EDC's case, that lysine-containing amino acid and that, or the amine containing amino acid and the carboxylic acid containing amino acids are going to be, 0 angstroms away from one another, because obviously dynamics matter, right? If you've got two alpha helices that are very, very close and very, very static, you'll get good cross-linking there. If they're quite far away and very static, you'll get less good cross-linking there. But two, pretty disordered regions can be quite far away from one another, and you can still get a lot of cross-linking there, because there might be an awful lot of flexibility within those regions. The other argument I have all of the time is you guys often do your measurements based off of AlphaFold, which is great. use AlphaFold, but if the cross-linking is saying something different to the AlphaFold, then that probably, means that the AlphaFold has some inaccuracies rather than the cross-linking, or you're using cryo-EM. Now, Chris Russo isn't in the room because I'd have to fight him if I say this, but maybe the cryo-EM isn't as accurate as you'd hope it was, especially when it comes to multiple conformations, right? We are trapping all of the conformations in one experiment here. Now, that can make your life a bit of a pain because you've got to try and break down what different conformers are present, but we are seeing all of the conformers in one experiment. Oftentimes in cryo-EM processing, unless you specifically are looking for multiple conformers, you keep one, lose another, and carry on with your experiment, right? That isn't what's going to be happening here. The reason I use this as an example is crosslinker solubility and permeability. If you've got a crosslinker that won't go through a membrane and you're looking at things that are inside a membrane, that won't work. It's pretty self-explanatory, and vice versa. And then sample complexity, just at the end here. When we run a mass spectrometer, it can only do a certain number of scans per minute. We try and balance mass spectrometry time with complexity of samples, but there's obviously a limit there, as well as what we can load onto columns and things like that. So essentially, the number of crosslinks does increase with sample of complexity, but it's not a linear increase in the number of crosslinks we get per protein. So the dimer experiment up here, there might have been like 600 crosslinks in that experiment between two proteins, so like 300 crosslinks per protein. And the one down here, I think when I did count it was about 25 proteins. That had about 2,500 crosslinks in, so averaging about 100 crosslinks per protein. So we tend to get less dense crosslinks as the sample gets more and more complex. So this is the bit that you're probably more interested in. It's sort of a protocol for crosslinking. Get stuck in. It's not as tricky as it might look. And if you're making really nice pure complexes, just go for it. Send me an email. We can work things out. This is what I added in last night as just some like useful tips for what we're actually looking for. But essentially, when you do your cross-linking itself, make sure it's all low bind. We have real issues. I know most people would use low bind anyway, but if you're doing cross-linking in vessels that aren't low bind, we do get a lot of adhesion to the side. You can't use amine containing buffers. I think that's quite obvious. If you're using NHS esters and you've got a load of tris in there, it's just going to react to all of your cross-linker up, and you're just going to get a really horrible mix of peptides react to tris or just nothing at all. Keep all your reagents really cold when you're doing your cross-linking reactions. You are adding quite a lot of cross-linker in as a volume. You just want to make sure that your complexes stay together. It's really important to make sure that your complex is stable with the cross-linker in, and I'm going to show you how we tend to look at that. Then when you're finished with all of this, quench it all to make sure that your reaction just doesn't continue and continue and continue, so you get a lovely blob of insoluble cross-linked together complexes on complexes. And then drop quench samples off a mass spec and talk to Thomas. And if you want to do this, talk to me. We can chat about best ways to do things for your precise complex. The ones on the left is just what I did originally, but essentially it's really important that your sample is as clean and as pure as possible. That's not to say we can't do cross-linking with highly complex samples, but if you're only interested in one specific complex and you bring me something that contains tens of different complexes, then you're going to get less information about the complex you're interested in. I can only load a column and a mass spec with so much material. The more of it that is what you're interested in, the more information that you'll get out of it. Better data, you always get better data if you understand your sample well. So it's probably worth starting with normal, non-cross-linked pull-down experiments. If you don't know what proteins are present, do some mass spec beforehand, get to know what proteins are present, and then we can start doing cross-linking experiments. Now, that's not to say we can't find unknown binders with cross-linking. We can, and we do that a lot. But I have had quite a few people just drop off samples and tell me they don't have a clue what's in there. That makes doing cross-linking incredibly difficult when it comes to the searching at the end. So the best way to make sure that your proteins are actually cross-linking or actually working is just run a gel of them. So you don't need to use too much protein to do this. but essentially run your cross-linking at a few different concentrations. I'll get into that in a bit. But what you'll see is initially, you'll see, hopefully without any cross-linker in there, you just see monomeric protein, just lone protein that comes out. And as you increase your cross-linker concentration, you see things shift up within your gel. They're getting heavier, they're cross-linking together. What you've seen right on the far end here is if you see a load of protein that's stuck in the well, that means you've over cross-linked. Essentially, you've got a load of non-specific interactions starting to occur between complexes. We can digest that and have a look. It's pretty pointless though. You end up getting a lot of cross-links that don't make any sense because you've just got complexes non-specifically interacting and just clumping together. So what we're looking for is sort of somewhere between these two, in that you've got lots of sort of complex, intact complex, lots of cross-links that are going together. What it actually tends to look like is more something like this. You obviously don't get like perfect cross-linking of everything with one particular cross-link to one other thing. You get a bit of a mixture. So everything gets very fuzzy. And essentially this one here is over cross-linked. This is probably the concentration that we go for when it comes to the cross-linking. Yeah, sort of this smeary thing in the middle. That'll have a good mixture of sort of dense cross-linked, less cross-linked, and then we should get some quite good information out of that. But yes, that's important to remember. If you do want to do this, I've now essentially, we get everyone to just quickly check on a gel beforehand that they've actually got firstly, they've got cross-linked complex, and secondly, they don't have the one on the end here, which is just completely over cross-linked complex. That was completely useless. Roughly when it comes to how much sample we need, this is changing as we get better instrumentation, but as a very, very approximate measure, and come talk to me if you are particularly worried about sample concentrations and things like that. That's roughly what we ask for. So sort of 50 to 100 micrograms of samples for those bigger complexes, and for smaller dimers, stuff like that, we can probably even go lower than 20 micrograms, but that's roughly what I ask for within those regions there. So that's just worth bearing in mind. And then cross-linker concentration is kind of in those regions that I tend to recommend people start at. So sort of 4 or 5 different concentrations between those two numbers should get you a gel that looks roughly like this. If it doesn't, come talk to me and we can have a chat about what we think that might be. But before you do that, do make sure you're not cross-linking interests. I've had so many people come down and say their cross-linking isn't working, and they've been cross-linking interests the whole time. So yes, that's sort of what we want to look like when it comes to cross-linking itself. When it comes to what cross-linkers to use, there's 2 really common ones when it comes to the NHS esters or di-NHS esters. We've got FOX and DSSO or DSBU. These are both super simple for me to do the processing of. So if you want to, they're not particularly expensive either. So if you want to buy some of these and start mucking around with cross-linking, please do. And if you bring them downstairs, I won't be angry that you've used something weird and wacky that I'm going to need to change all of my data processing for. So that's totally fine. The one on the end there is SDA. Now that's a bit different. So what we've got here is the standard NHS ester, like the other ones. So this comes in and reacts with a lysine. But then what we've got on the end here is a diazarine, and that's UV active, forms a radical, and that then can react with any amino acids. So this has got a bit of interest recently, and we're getting more and more people to use this. We've got a little box downstairs that the workshop made for us that essentially has a load of UV LEDs that gets us really efficient UV cross-linking. But essentially, you just do exactly the same thing that you would normally do is you incubate your cross-linker with your protein complex, and then in about sort of a few milliseconds of UV activation, the diazarine comes off, or the nitrogen comes off the diazarine, and you end up getting a radical reaction to a protein. Now, this does have a slightly useful thing in that because it's a lysine to any amino acid cross-linker and it happens so quickly, we get dense cross-linking. We also, there are papers that are suggesting we get what could maybe be described as more accurate cross-linking, which is this idea that because you're trapping the states much quicker, that you get sort of more controlled cross-linking across the interaction interfaces. So it looks something like that. So essentially, you don't need a lysine on the other side. It could be literally anything else. So that's quite interesting. Something else that we can do, especially with SDA, because we tend to track the complexes a bit better, is that we can start to think about conformers. So when we're looking at where proteins or chains within proteins might sit, we might get something that looks like that. And what we can say there is, well, the one on the right-hand side, those crosslinks seem to make sense. We've got two alpha helices that are in close proximity and they're interacting. These ones make less sense, right? It probably looks, instead of the thing on the left, it probably looks more like the thing on the right, because those two alpha helices are much closer together. Now, this is pretty weak because we've done this by eye and we're just sort of making some assumptions here. But there was a paper that came out in 2023, which is by Yuri Ravsila, and this is something we're using a lot more now, which is essentially an AlphaFold plugin that's called AlphaLink. And this is now getting a lot of use, and Sammy's done quite a few things with this. Sammy Shaban from the class group, who does a lot of AlphaFold stuff. And essentially what we've got is we can take these forbidden overlength, however you want to describe them, crosslinks from an AlphaFold prediction. We can then put those in as restrictions. So it can't, we just tell it that that's impossible. It can't be that far out. And then AlphaLink essentially then does another AlphaFold prediction, but forcing those things closer together and sees what the best match is. And it works pretty well, actually. We're still getting to grips with some of the things of it, but it sometimes gives you some very, very strange data, but often it gives you some very, very nice data, and it can really help with AlphaFold predictions, especially when you get strange regions, especially in AlphaFold 3, when you can get some really strange regions where it's sort of, it places them in places, doesn't it? And this is a way for us to sort of pull some of that information back. So that's something that cross-linking is very useful for, and something we're starting to use a lot more of. So that's kind of the bare bones of what we do for cross-linking. I'm now going to talk about some stuff that we're sort of working to develop in the lab. Now, just because we're developing it certainly doesn't mean we can, like, people in the room can't get involved with this work. Actually, we need you guys to give us samples a lot of the time. So something that we're really focusing on at the moment is in-cell cross-linking. So almost all of the cross-linking we do is currently from recombinant proteins or from pull down, essentially proteins that have been purified in some other way prior to cross-linking. What we're looking to try and do is capture proteins within their most native states. And we've got very, very similar considerations to take into account, but it's much, 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 much more complex. We are then, our search space becomes that crazy high, stupid number of literally millions of proteins, potentially. Currently, we're doing some funky things where we look into cell fixation, partial permeabilization, followed by cross-linking. And that gets us currently not very good maps. We've got some data now that's a bit better than this. This was from a little while ago. But this is an archaeal cell that we essentially know nothing about. And we're starting to pull out some protein complexes from that. I've actually got about 3 of these data sets processing at the moment, which hopefully give us even more cross-links than that. But essentially, if we don't know anything about it at all, this is a way for us to generate some interaction data. So if you think you might be interested in doing in-cell cross-linking, do come talk to me. That's something that we're starting to do, but we're miles away from having a complete interactome from one cross-linking experiment. So don't expect that, but we might be able to get you some interesting data. Also, we can do in-cell cross-linking and then pull down on that protein, which we have very mixed results with, but some of those have been really successful. So come talk to us if you're interested in doing that. Happy to try and facilitate something there. So essentially, just a review. Just try it out. Just genuinely just try it out. If you've got a pure protein complex and you haven't run cross-linking data of it, and you guys love to spend 3 years trying to get a good cryo-EM, we can get a cross-linking data set in like a week. So just try out the cross-linking data set. It will look really good in your first year talk, hint, and then get back on with your cryo-EM. So yes, check with gels, talk to me or Cats before submitting samples and try it out. So that's the cross-linking bit of the talk done. Now there is something else. This is when we stop talking about science and it becomes a political rally. Okay, intact protein mass spectrometry. Now, hardly anyone does this and it drives me nuts. Right. How many times has your gel band looked weird? is your protein exactly what you think it is? Like, genuinely, have you ever checked that you're making the right protein? I know you guys love gel bands. I know you do, but have you actually ever stopped to check you're making the right protein? Have you ever thought about what PTMs might be on your protein? I used to work in pharmaceuticals, and we would spend like 2 years looking at one protein and every potential PTM that could be attached to that protein, because we knew that that completely changed the properties of the protein. Yet you guys will run cryo-EMs all day and not factor in PTMs into it, which I find mental. I love it. It's brave, but I don't understand it at all. Right. It's a super quick way to confirm that you have made the right protein that you've probably never. Oh no, that's my phone alarm going off to remind me of this. If you need to do that, that's like a really super quick way of doing it. And the answer to this is intact protein mass spectrometry. And I'm not joking. Have you actually seriously looked at that you're making the right proteins, that the right proteins are in your complexes? Seriously not joking. Have you actually ever checked that gel band? People do gel bands for years and they'll never check that they've actually made the right. Gel bands suck, okay? They suck. Western blotting is also not infallible. People will be like, I've never checked it, but I've got an antibody, and it kills me. It kills me. They will spend 2 years and they'll never check a protein, right? It's insane. Okay, that's enough about the political side of the run. But essentially, we've now got a really cool new instrument that can turn around protein samples really, really quick. It's very accurate, or it's very precise in that we get measurements to 4 decimal places, and then we get protein accuracy within 5 parts per million, which is accuracy to about 1 or 2 decimal places for the average protein. So this isn't mass phytometry that gives you these wacky lumps. I'm sorry, Stephen, but it gives you these wacky lumps that are about 200 kilodaltons in width. This is something that gives you 2 decimal places of mass accuracy for your protein. Now, you just bring it down in a little tube and we can run this in about half an hour. If it's got PTMs on it, we can tell you what PTMs are present. I'd be lying if I said it's quantitative, but it's reasonably quantitative. We can do reasonably good studies in looking at this. So if you want to see how much phosphorylation that kinase is actually doing to your protein, just bring it down in an Effendorf. We can run it in 30 minutes, tell you that it's actually phosphorylated or not, tell you how much phosphorylation is there, tell you if it's all on one protein or spread across multiple different proteins. We can look into stuff like that super quickly. Just bring it down. Yeah, one of the best ways to assess PTM. The other thing we can do on this, it's a bit trickier, but come talk to me, is we can actually look at native mass spectrometry. So we don't even need to look at individual proteins. We can put an entire complex onto the instrument. For those of you that have ever read something from Carol Robinson, this is what she's based an entire research career off of. It's incredibly useful. But essentially, we just take your protein complex and we run it as one intact mass. So then we can start looking at things like stoichiometry and stuff like that. And, then we can do the same thing on the same complex, but in a denatured mode, and we can look at what individual proteins are present there. And then we can actually start to get a good idea of what your entire complex is. We've got an Ascend now, which is a much more powerful mass spectrometer, where we're actually hoping to do this in sort of a different order, which is we spray in native mode, we get a really nice native mass, and then we start activating your protein to break it down into the individual protein constituents. So this is a way we can not only look at stoichiometry, but we can actually start to understand what proteins are present at what stoichiometries. And we can also, this is a bit of a, not quite yet, but we're working on it, and other people are working on it, which is you can look at how this breaks down to get an idea of what's interacting with what as well. Frank Sobot from Leeds, if you want to Google that, he's just done a really, really interesting study into looking at how we can fragment proteins in ways to look at what sites are interacting with what and what proteins are interacting with what other proteins. So that's something we're also looking into to start doing. So do come talk to us if you think you've got interesting things that you might want to look at. The other thing that we do, we've just done a load of this with Roger Williams's group, which is they have non-covalent interactions of various phosphorylated species with larger protein complexes. So if you can have 2, 3, 4 of these sort of phosphorylated, it's like small phosphorylated peptides that are doing the interacting. We can see those non-covalent interactions using native MS, which is super interesting, and you can't do that in gels or anything like that. And how they were previously doing it is that they were trapping and then releasing these peptides and just seeing how many they've got, but that wasn't telling them if it was sort of evenly spaced across different sites, or how that was looking when it actually came to their protein. So we can get things like that. So non-covalently bound small molecules can also be done by native mass spectrometry. So that's something we're running a lot more of now as well. So please do come talk to us if you think either of those techniques, well, intact mass is just useful to everyone, and it takes 15 minutes, and you literally don't need to do anything. Just drop it off. So long as it's not in detergent, we can run it. So just drop it off whenever you want. And essentially, that's my that's me done. So thank you. Come talk to any of us, but obviously, if you're interested in cross-linking, come talk to Kat or me, or Intact, come talk to me. Kat doesn't really care. We can run it so quickly and so easily. Just bring it down and come find me, and we'll run it there and then. Cool. That's everything. Thank you very much. Are there any questions? Yes.
How much protein do you need for the intact mass spec?
Roughly sort of one. Yeah, that would be useful to put on the slides. Roughly 1 to 2 micrograms. It's not very much at all. Yes, if it depends on obviously how complex that sample is, right? So if you've got one nice pure protein, like 1 microgram is more than enough. If you think it might be a bit of a mess of stuff, then bring maybe 5-10 micrograms down. We do a larger injection and then get more signal per protein, right? So, but yes, yeah, it's not very much. Real. Cheers. Thank you very much.